Hello, my name is Veronique. I'm a scientist by training, and I've been working in the group of Dr. Bonneman for 10 years, working on developing molecular therapies, in particular for the collagen VI dystrophies. Today, I will discuss two topics. The first one is the state of our research on an exon-skipping therapy for the Pseudoexon variant. And the second topic are efforts to identify a viral vector to deliver molecular therapies to the collagen VI expressing cells. So the first topic, as you know, the pseudoexon is one of the main types of dominant negative variants. It occurs in the COL6 alpha-1 gene, and the ideal therapeutic strategy for this variant is an exon skipping. And this can be achieved with antisense oligonucleotides, as I will show. First, I want to illustrate how the pseudoexon variant happened so that we all understand why is exon therapy a good strategy. So this is the simplification of a gene. It's composed of DNA. So when a gene is expressed, first, the exon and introns are copied into one long single molecule, which is a precursor to the messenger RNA. Then the introns are removed through a process called splicing, which leaves the messenger RNA or mRNA, and these are the instructions to make a protein. So what happens in the case of the pseudoexon? Well, as you may know, the pseudoexon is caused by an intronic variant, a single nucleotide chain in the DNA. So this is illustrated here with this red star. So you may think, why does an intronic variant matter since the introns are removed? So here's why. When the gene is expressed, the variant in the intron tricks the cell into thinking that there is an exon hidden inside this intron. And during splicing, it leaves behind a little piece of the intron that we call pseudoexon. And this information is passed on to the protein, such as the protein now has a different composition and structure. So now we can block this pseudoexon with antisense oligonucleotides. Antisense oligonucleotides are short sequences of DNA or RNA, and we designed them to recognize sequences within the intron. And the idea is that if the antisense oligonucleotide can bind to the precursor mRNA before splicing, it can force out the pseudoexon, leaving the native sequence of the messenger RNA and the instruction to produce the native protein. So this strategy of exon skipping or splice modulation in general has been successful for other conditions, namely Duchenne muscular dystrophy and spinal muscular atrophy, with some products already on the market for those conditions. So now here's a summary of the work we've done so far. We've designed several antisense oligonucleotides to recognize different parts of the pseudoexon sequence, and we've tested them in cultured cells. So as you can see, two of these antisense oligos are effective at blocking the pseudoexon. So showing that they are affecting at pushing out the pseudoexon during splicing. Other collaborators of ours, the laboratories of Dr. Muntoni and Zhao at UCL, have also shown with different chemistry that antisense oligonucleotides are effective at blocking the pseudoexon. So that's on the mRNA level. Now, what happens at the protein level? So we looked here in proteins that we immobilize on a gel, and we look at their abundance with an antibody. So this is an antibody that recognizes specifically the protein that was formed with the pseudoexon. So of course, control cells don't produce this protein, but patient cells do. But after treatment with antisense oligonucleotides, the abundance of this protein is greatly reduced. So this is a good sign that the antisense oligo are able to block the pseudoexon and to reduce the amount of the protein with pseudoexon produced. So next, we looked at the matrix that is produced by those cells. So in patient cells, the matrix is interrupted because of the abnormal structure of the protein. But with antisense oligonucleotide treatment, the formation of long microfibrillar structure was restored. So showing that we can improve the morphology of the matrix with antisense oligonucleotide treatment. So next, we decided to create a mouse model. So here we took the mouse COL6A1 gene, and we replaced a segment of the DNA for the human sequence. And we created two different types of mouse. One that contains the reference or the native sequence that we called UMC, and one that contains the intronic variant that causes the pseudoexon, which we call UMT. And so first we looked in the skeletal muscle at the mRNA level in these animals. First, all of the animals splices the human exons properly. But in addition, in the UMT animals, the pseudoexon is expressed. So that shows that it's a good model to mimic the situation in patient muscles. Next, we looked at the muscle function of these animals. So we measure their grip strength with this small apparatus. And we found that UMT animals have lower grip strength. So these are males, these are females. So suggesting that they have a muscle weakness in their grip. Next, we looked at the histology level in different skeletal muscles, and we found that UMT animals have higher number of central nucleated fibers, which are signs of degeneration and regeneration of the muscle. So all these signs point to a model where there are signs of muscle weakness. Okay, so to summarize what we have here, we've made the proof of principle that the pseudoexon can be skipped. And not only that, that skipping it improves the protein. We have generated an animal model. The animal model expresses the pseudoexon, just like it's expressed in patient muscles. And the animals show signs of muscle weakness. So this validates this model as a good in vivo model to test our therapies. So what we need next, we need an antisense oligonucleotide chemistry that will deliver itself to collagen VI producing cells in muscle. And for that, we are working with different pharmaceutical companies to test different chemistries. Or as an alternative, we could use a viral vector to deliver a cassette that expresses a small antisense RNA. And this brings me to my next point. A delivery vector for small antisense RNAs. So antisense oligonucleotides are not the only modality to perform exon skipping. We can use a vector that expresses small nuclear RNA that can also perform exon skipping. But not only that, this vector can also expresses microRNAs for allele-specific silencing or guide RNA for gene editing, such that a viral vector could be applied to a whole host of antisense RNA for different strategies. In gene therapy field, the adeno-associated viral vectors are the vectors of predilection. These are derived from the adeno-associated viruses, or AAV. These viruses are non-pathogenic to humans, and they are composed of a capsid, which is a shell of proteins that encapsulates a single-stranded DNA genome. So how do we make a virus into a vector? Well, we remove the viral genome and we replace it with an expression cassette of interest. So as I was mentioning, we can express small nuclear RNA to mediate exon skipping. We can express microRNA for allele-specific silencing, etc. So the important thing is that this virus is capable of reaching and transducing the cells that express collagen 6 in the muscle. So for that work, we decided to work with a reporter mouse model. This mouse model expresses a green fluorescent protein into the COL6 expressing cells. So if you can see here on a biopsy from these mice, all the little green dots here are cells that express collagen 6. So the green fluorescent protein allow us to locate and to isolate these cells in particular. We partnered with the lab of Luc van der Berg in Boston. They have created several AAV libraries, and we started working with one of their libraries that contained 2,048 different AAVs. Each of them is barcoded with a unique barcode. So we injected this library into our reporter model, waited 1 month, harvested skeletal muscles, and used a technique called fluorescence activated cell sorting to specifically isolate the cells expressing the green fluorescent protein, and by consequence, the cells that express collagen 6. And we isolated the DNA and sequenced the barcodes present inside these cells. So this allows us to find which of the variants infected the cells of interest. So this is a work in progress. We're currently finishing up a pilot study for this project. Next, we will extend the study to a larger number of animals and also to a larger number of libraries. Once we find a few positive hits, we're going to validate them in vivo in the Pseudoexon mouse model, for example, for their ability to deliver antisense molecules to the collagen 6 expressing cells. So this is a work in progress, and I hope we'll have data to show in the next year. So I want to thank my colleagues in the Bonneman lab, in particular Astrid, Fadi, Lindsay, and Ying, our collaborators in London, in Boston, of course, the Foundation Noelia and Cure CMD for their unwavering support for our different projects. MD UK supported us for the creation of the Pseudoexon mouse model, MDA, CIHR, and FRSQ for fellowship support while I was working on the Pseudoexon variant. So thank you.